Welcome back. So here we are, week 4, module 4, and now we're getting to cell structure and function. So cytology, which is the study of the cell. And once again, just like we did for the last 2 modules, we're going to take one chapter and really delve into it a bit deeper and spread it out over 2 modules. So for both this week and next week, we're covering cell structure and function. So to start off, we're going to talk about what is the cell and the basic principles of the cell theory. And basically the cell theory is how all living things are composed of cells. We're going to look at how the size of the cell is very important and how the surface area to volume ratio limits cell size. That's why cells don't get really big. We're also going to talk about how we study cells using microscopy, using a microscope, and how that's utilized. And then we're going to start delving into how cells are organized. So we're going to talk about the composition, the basic composition of a human cell, once again studying human biology here, and basically state their basic structure, function. We'll get even more into that in the next module, next week. We're going to look at the differences between a eukaryotic cell, which is what human cells are, the complex cells, versus a prokaryotic cell, which is a very simple cell. Simple single cellular organisms. And we're going to talk about how eukaryotic cells, these complex cells, actually developed from these simple prokaryotic cells. So we have the simple cells, we're going to compare and contrast to the complex ones, but we're going to see how these complex, for example, plant and animal cells, originated from those simpler prokaryotic cells. So once again, we're still just talking about cell organization at this time. Then we're going to go ahead and get into the plasma membrane. Now remember last week, we did talk about lipids, and specifically phospholipids, how they're the foundation for all the membranes in your cells, the plasma membrane and the membrane-bound organelles. Well, now we're going to really go deeper into that plasma membrane. We're going to look at that phospholipid bilayer and all the other molecules that also cross it, like those complex proteins, because remember, proteins are cool. They create the machinery of the cell for the most part. So we're going to look at the proteins, we're going to look at how cholesterol enters it, we're going to look at even carbohydrates that are in there, the carbohydrate chains that are connected to certain proteins within that phospholipid bilayer. So we're going to talk about the basic structure of that cell membrane and all those different molecules that do actually make it up. We're going to look at how different ways of water and different ways other elements can cross that membrane, whether it be through diffusion, osmosis, facilitated diffusion. We're going to be defining all of these terms and state the role of each of them as far as water and different components entering and exiting the cell. We're going to talk about when the cell is in a solution, whether it be what's called an isotonic solution, hypotonic solution, or a hypertonic solution, basically whether water is going to want to flow in or out of the cell. This whole concept is going to be called tonicity. You can tell that by the tonic here, right, on each one of those. So we're going to talk about this concept of tonicity and how it relates to the direction of water flow across a membrane. We're also going to compare what's called passive and active transport mechanisms. Passive not requiring energy, active requiring energy. So once again, the membrane is meant to separate 2 solutions, an outside aqueous solution and an inside aqueous solution. How different ions and molecules get across has to do with these membranes that cross it. What are they going to allow in, and does it require energy? So if it requires energy to allow things to be pumped in or out, well, then it's active transport. If it doesn't require energy, then it's passive transport. So once again, we'll dig in a little bit deeper in comparing active versus passive transport. And then we're going to talk about how big molecules enter and exit the cell through phagocytosis, pinocytosis, and receptor-mediated endocytosis. So we're going to talk about how these complex eukaryotic cells, those are the complex ones that make up the human body, can move large molecules across the membrane either in or out of the cell. So talking about the cell, the basic organization, and then delving into that plasma membrane and how substances cross it. Let's get started.